Hello, this is Mohammad, and here I'm presenting my research on Pseudomonas aeruginosa acyl-CoA dehydrogenase fatty 2 and how it affects our pathway, mitochondrial unfolded protein response, and how it affects bacterial survival during infection and also uh, host survival during infection. So we're using C. elegans as the host in Dr. Perugino's lab. So first of all, when worms are infected with Pseudomonas aeruginosa, they activate MITO-UPR in the host. So MITO-UPR is activated and it makes sense because bacteria, Pseudomonas aeruginosa, produces toxin that target mitochondria, and UPR is activated to protect mitochondria and alleviate stress in the host. So on the first day, we have activation of the UPR, but surprisingly, on the second day, we don't have that activation anymore. UPR is repressed in the worms that are infected with Pseudomonas aeruginosa. We have toxins, we have everything, but UPRs is repressed. Previously, we showed that a loss of function in a gene called FADI2 can result in sustained activation of the UPR during the infection. And worms that are infected with the FADI2 knockout bacteria, Pseudomonas aeruginosa, can survive much longer. So what is this fatty 2? Fatty 2 is a metabolic enzyme, is in a silk-weight dehydrogenases among different silk-weight dehydrogenases involved in branching amino acid catabolism. So the first question we asked ourselves was that um, this is something specific to the fatty 2, or uh, this can happen, this suppression can this sustained activation of the UPR can happen if we mutate any of those other acyl-CoA dehydrogenases. And our results showed that only uh, FADI2 is involved in repression of the MITO-UPR in the host. So only loss of function mutation in the FADI2 in the bacteria can result in the sustained activation of the UPR in the host. So here we started looking at the bacterial environment. So this is a very stressful environment inside the lumen of the C. elegans. Bacteria is experiencing during infection, full of different forms of stress: acid stress, heat stress, osmotic stress, oxidative stress. And we started looking at fatty 2 knockout and the vital form of the bacteria. Uh, we started looking at their growth under each of these stress conditions. And we found out only under oxidative stress, there is a significant difference between the fatty 2 knockout and the wild type. So the wild type that has the fatty 2 inside the bacteria can survive much better under oxidative stress. What does that mean? So, first of all, let's talk a little bit about how oxidative stress, for example, let's say one hydrogen peroxide, can alter proteins' function. So, cysteine sites in proteins can be sulfonylated, and they can react with, for example, hydrogen peroxide and be sulfonylated, and this sulfonylation can alter the function of the protein. So, interestingly, fatty 2 has two conserved cysteine sites that have the potential to be sulfonylated. We replaced those ones with serine, and it's called the mutant that we constructed, Pseudomonas aeruginosa C1, C2, and we looked at the bacterial growth under the oxidative stress compared it to the wild type. So the white type survived much better. It showed the same growth as the fatty 2 knockout. And we looked at the induction, again, same induction as the fatty 2 knockout in fatty 2 C1, C2. Even worms that are infected with the uh, Pseudomonas aeruginosa C1, C2 survive much better than the ones that were in fact infected with the Pseudomonas aeruginosa wild type. So altogether, these systems that can play an important role in activation of the fatty 2. A fatty 2 might be activated directly by oxidative stress to protect the bacteria against that oxidative stress.